So, plant growth regulators are also addressed as plant hormones or phytohormones. Plant hormones or phytohormones. What are these? Are they any chemical substances or are they any inorganic elements? Are they any organic compounds? What are they? Yeah, they are present in plants, but what are they composed of? They are small, simple, organic chemical constituents, okay, secreted from any part of the plant body, okay. They are small, simple, organic chemical constituents. Synthesized from any part of plant body, okay, and their function can be at the part of their synthesis or any other part of the plant body. For example, if I have to take auxin, auxin is known as rooting hormone, okay. It is synthesized most at the shoot apical meristem, but its most of the function is at what region? Root region. So, that is the reason it is called as rooting hormone. Same the case if you observe with cytokinin. Cytokinin is addressed as shooting hormone. Higher amount of cytokinin synthesized at root apical meristem, but as its function is more at the shoot apical meristem, it is known as shooting hormone. So, it is synthesized at any part of the plant body. Its physiological function can be at the part of their synthesis or any other region of the plant body. Clear with the definition of plant growth regulators. Regulator means what ma? It can promote or it can inhibit the activity of the plant. It can promote the growth or inhibit the growth. Okay. As such, how many plant hormones we need to learn? There are so many amount of plant hormones, but here we are learning the basic first derived or discovered phytohormones. Those are what ma? Auxin. First we will learn plant growth promoters. And then plant growth inhibitors. Plant growth promoters include auxin, cytokinins, cytokinins and gibberellic acid. Plant growth inhibitors are only one, that is abscisic acid, which is also known as a stress hormone or anti-transparent hormone. If you consider ethylene, ethylene is a gaseous hormone which plays dual role as plant growth promoter and as plant growth inhibitor. Clear till here? Any doubts anywhere ma? Now, at this point only you have to be clear on what you have to learn from phytohormones. With respect to each phytohormone, you have to be clear on the discovery. First thing is what ma? Discovery. Second is precursor. Third, derivatives and types, or you can say classification of that particular phytohormone. And fourth is its physiological functions. Followed by applications or uses. These are the 5 areas you have to focus on each phytohormone, from which you will get the questionnaire as well. Okay, ma. Now, first table itself I am making you it clear about the derivatives and precursors. Can everybody see this? First, auxin. What is the precursor of auxin? Tryptophan. What is meant by precursor? The starter to synthesize this phytohormone. In the complete biosynthetic pathway to synthesize the phytohormone, what is the starting component? Okay. For auxin synthesis, what is the precursor? Tryptophan. And auxin phytohormone are derivatives of what? Indole compounds. They are derivatives, indole derivatives. So, what is the precursor beta? Tryptophan for auxin. What are its derivatives? Indole derivatives. Remember, cytokinin we will learn natural cytokinin and artificial cytokinin. Natural cytokinin is zeatin, artificial cytokinin is kinetin. And its precursor is adenine. Okay, usually these are purine derivatives, more specific is adenine. Derivatives are also adenine derivatives. Next, gibberellic acid, precursor is acetyl coenzyme A or mevalonic acid, and derivatives are terpenes, terpene derivatives they are. Next, abscisic acid, which is plant growth inhibitor. Precursor is violoxanthin, and they are the derivatives sesquiterpenoid derivatives, means it is having 15 carbon terpenoid. Next, ethylene if you see, which is a gaseous hormone, precursor is what ma? Methionine, and it is a gaseous hormone composed of carbons and hydrogen, that is it. First you take down this table, I will tell you how you have to remember it easily. From precursors and derivatives you can see 2 to 3 PYQs of NEET. Take down carefully ma. Done, right? Now you look at here. First is auxin. What is the precursor for auxin? Tryptophan. Be louder. What is its chemical composition? Cytokinin if you see. Okay. The natural form of cytokinin is? Artificial form is? Simple thing you have to remember that adenine is the precursor and they are adenine derivatives itself. Okay. adenine is the precursor, adenine are derivatives. For auxin, tryptophan is the precursor and they are derivatives of indole compounds. Next comes the gibberellic acid. What is its precursors? It can be acetyl-CoA or mevalonic acid. Derivatives, terpene derivatives. Next, abscisic acid. Its first precursor is violaxanthin. Derivatives, you just remember terpenoids. Okay. Precursor is violaxanthin. Ethylene, methionine is the precursor and derivatives include gaseous hydrocarbons. That's it. Understood, right? Now we will move on to the discovery of auxin. First phytohormone, auxin, its discovery. Two types of coleoptiles you will observe. First, you have to be clear on what is a coleoptile. What is a coleoptile? If I have to consider a maize grain, okay, this is the kernel, right? Here you will see the scutellum, fine, and this is the embryonal axis. This is coleoptile region, this is coleorhiza. Coleoptile and coleorhiza are the protective shields. You will see only in the monocot seeds, yes or no? You will not see coleoptile and coleorhiza in dicot seeds. So, coleoptile is taken from canary grass. Canary grass seedling. What is coleoptile? The extra sheath which is covering the plumule, small region above the plumule. Got it, ma? Okay. So, this canary grass seedlings, coleoptile is taken. First it is discovered by whom? Charles Darwin and his son Francis Darwin. What did they do? They have taken the coleoptile, okay, this is the coleoptile region. To this unilateral sunlight was flashed, okay, and after some period of time they have observed that there is a bent in the coleoptile region towards the sunlight. Bent is where? Towards the sunlight, which is showing what movement, beta? Phototrophic movement. Okay. Later this is isolated from oat seedlings, coleoptile of oat seedlings. By F.W. Wendt. What did he do? His experiment is different, okay. He has taken the coleoptile of oat seedlings and decapitated it. Decapitating means what? Taking off the apex of the oat seedlings, and then this decapitated coleoptile is placed over an agar block. Okay, when it is placed over an agar block, what happens? All the chemical constituents at this apical region were transferred into this agar block. After a certain period of time, this agar block was placed over the decapitated oat coleoptile, and this was exposed to unilateral sunlight. They have observed that there is a bent that is shown towards the sunlight. Okay. Once again I will repeat the experiment. See, the coleoptile of oat seedling was taken and it is decapitated. The removed coleoptile part is kept over what? Agar block. What is agar block? Which is a hydrocolloid where we have extracted it from red algae, right. So, over that hydrocolloid what is placed? The decapitated coleoptile of oat seedling, and it is remained like that for some period of time. What happens? The chemical constituents from this coleoptile were entered into this agar block. Then this agar block was placed over the decapitated oat seedling. Fine, right? Constituents from here have moved into the coleoptile and this caused a bent with when the sunlight is passed towards it. Clear, right? And the bent is towards the sunlight. Two things you have to be clear here. First part, who discovered the auxin? Okay, Charles Darwin and Francis Darwin from coleoptile of canary grass. Then who isolated it? F.W. Went isolated it from coleoptile of oat seedlings. He has isolated it from coleoptile of oat seedlings. And auxin is isolated from human urine as well by F.W. Went. Clear, right? Discovery is this scientist and the example is coleoptile of canary grass. Isolation by F.W. Went, oat seedling. Clear with the discovery of auxin? Now, auxin classification. You are taking this? Take it parallelly at the class time only, ma. Just take down those 2 points where statements can be stressed, who discovered, who isolated. And auxin is the first phytohormone to discover. Auxin classification also you listen. Auxin is classified into 2 types based on whether it is natural auxin or synthetic auxin. Natural auxin can be indole acetic acid, indole butyric acid. Synthetic auxins include naphthalene acetic acid, 2,4-dichlorophenolic acid, 2,4,5-trichlorophenolic acid. Clear? These are the natural and synthetic auxins. If you are done with this, we will move on to the physiological functions of auxins and applications or uses of auxins. Functions. Yeah. First major function of auxin is, we have discussed, auxin is majorly present at apical regions, most at shoot apical region. So, the auxin constituent at shoot apical meristem might be, root apical meristem might be. This will dominate the growth of lateral buds. Okay, this will dominate the growth of lateral buds. In simple terms, it is inhibiting the growth of lateral buds. It will promote the growth of apical buds and inhibit the growth of lateral buds, which is known as apical dominance. Clear with the first function? Second function, auxin promotes cell division not more than cytokinin. Okay, when you consider cell division, auxin and cytokinin are synergistic in their function. Okay, wherever I say synergistic and antagonistic, please focus there, because you will get the questions on synergistic and antagonistic functions of phytohormones. Okay, synergistic means 2 phytohormones promoting same function. Antagonistic means function which is inhibited by one hormone, promoted by the other. Okay, so here if you see cell division, okay, it is promoted both by auxin and cytokinin. Cytokinin promotes more than auxin. So, here what are synergistic in function? Yes, synergistic auxin and cytokinin. Okay. Next, abscission. What is abscission, ma? Fall of leaves, fruits is known as abscission. Auxin will prevent the abscission of younger leaves and younger fruits, and it will promote the abscission of older leaves and senescent parts of the plant. Okay. Next is xylem differentiation. Auxin promotes xylem differentiation. Xylem includes how many tissues inside it in an angiosperm? Xylem is a complex tissue, yes or no? It includes what? Tracheids, vessels, xylem fibers and xylem parenchyma. Auxin helps in what? Xylem differentiation. Clear? Clear with the functions of auxins. Okay. Now, how auxins are applied, how they are used, we will learn. See, auxin will promote rooting in the stem cutting regions. For example, you have cut some stems. To promote rooting in the stem cutting regions, that is to promote adventitious root formation over the stem cutting regions, auxin will promote that, rooting in stem cutting regions. Next is flowering. Okay. Rooting is promoted by naphthalene acetic acid and indole butyric acid. Okay. Flowering in the plants, when auxin is sprayed, flowering is seen. Okay. Flowering is promoted. For that, what type of auxins are used? Naphthalene acetic acid and 2,4-dichlorophenoxyacetic acid. These are the auxins that are used. To promote parthenocarpy, these days everywhere we are getting seedless fruits, be it banana or any other fruit, grapes, okay, any other fruit we are getting it seedless. That is because of parthenocarpy. How are the fruits formed without seeds? By spraying auxins. What type of auxins are sprayed? Indole acetic acid and indole butyric acid. Next, weedicides. Okay. When 2,4-dichlorophenoxyacetic acid is sprayed over the lawns, that will give weed free lawns. Especially this will eliminate dicot weeds. This eliminates dicot weeds. Clear with all the functions and applications of auxin, ma? Yes. No, because it is inhibiting the abscission of younger leaves and flowers. If you see ethylene and abscisic acid, they will promote abscission based on the climatic conditions. See how it will happen is, any other doubt, ma? Clear? Shall we move on to cytokinins? How cytokinin is identified? it is the scientist Skoog. He is growing a tobacco callus in the test tube. Some undifferentiated mass. What is callus? Undifferentiated mass of cells. Okay. To this undifferentiated mass, when cytokinins are being applied, okay, there is an increase in cell division activity, and this is also showing differentiation, the tobacco callus. Okay. So, here comes what are the natural forms of cytokinins. The natural forms of cytokinins that is used. Natural form of cytokinin is zeatin, which is extracted from coconut milk, yeast extract, unripe maize grains, and kernels. Okay. Then it is Miller et al. Scientist Miller who has reported, we will address it as Miller et al. What is his discovery? He has isolated the artificial form of cytokinin in the form of kinetin from autoclaved herring sperm DNA. Okay. He has isolated kinetin. What is kinetin, ma? Synthetic form of cytokinin from autoclaved means what? Which is sterilized. Okay. From autoclaved herring sperm DNA. Okay. From a fish sperm, herring sperm is a fish. Okay. From its DNA, what is isolated? Synthetic form of cytokinin. You can also get kinetin from isolated form of tRNA. Why nucleotides? Because it is extracted from purine. Cytokinin is purine derivative. Yes or no? Auxin precursor. What is the precursor of auxin? Louder. Tryptophan. Derivatives? Indole derivatives. What is the precursor of cytokinin? Adenine. What derivatives? Adenine derivatives. Natural forms and artificial. Please remember. Important. Done, ma? Everyone? Now, what are its functions and applications? Cytokinin, the name itself is saying, cyto means what? Cell, kinesis means movement, division. Okay. So, here cytokinin is primarily involved in what? Cell division. When cells are dividing, they are more active, yes or no? More active cells will be moving more. Okay. They are mobile in their function. So, they actively participate in nutrient mobilization as well. Nutrient mobilization. Okay. Increase in cell division and nutrient mobilization. As it keeps the cells active, cytokinin will delay the process of senescence, which is explained in Richmond-Long effect. Fine. And this overcomes the apical dominance. For example, over a plant, if you spray cytokinins, what does it? yeah, it will promote the growth of lateral buds, whereby it will inhibit the growth of what? Apical bud. Okay, it will promote the growth of lateral buds and it will inhibit the growth of apical bud. This is used in tea plantations and hedge making. Okay, decapitation of the apical buds or spraying of cytokinins, this will be used in tea plantations and hedge making. And then growth of shoot, growth of leaves. These are the physiological functions of auxin. What is the synergistic function? Auxin and cytokinin cell division. About senescence we will learn. Okay, senescence is promoted by ethylene, inhibited by cytokinin. So, cytokinin and ethylene are synergistic in their function, sorry, antagonistic in their function. Okay, this delays what? Senescence. And with respect to apical dominance, which two hormones are antagonistic with each other, ma? Auxin and cytokinin are antagonistic to each other. Clear? Understood everyone? Next, in tissue culture, if you see the applications, if you spray cytokinins, that will obviously delay the process of senescence. In tissue culture, how you will apply the cytokinins? If auxins and cytokinins are given in equal ratio, that will help in the formation of callus. Okay. If auxin ratio is more than cytokinin, that will first initiate what? Rooting. If cytokinin ratio is more than auxin, that will first initiate what? Shooting. Understood, ma, everyone? Synergistic, antagonistic activities, applications. Done? Next is gibberellins or gibberellic acid. How is this discovered? This is discovered by the scientist Kurosawa. How did he discover? In Japan, once they have noticed that the rice seedlings are growing abnormally too tall. Okay, rice plants are growing too tall. That is because of what? By a fungal infection. The fungi name is what? Gibberella fujicuroi. Okay, this is the fungus. This is infecting the plant, so the plant is growing too tall than the normal rice plants. This plant is abnormally tall, these rice plants, because of the fungal infection. What are the chemicals that are causing the effect from this fungus? When those are examined, those are what? Gibberellins. Gibberellins. Gibberellins are responsible for the elongation of the plant, is what discovered. As such, some hundreds of gibberellins were discovered. Okay, what is the nature of the gibberellic acid? It is acidic in nature. Okay, hundreds of gibberellic acid is discovered, of which gibberellic acid 3 is widely used. Just you have to remember, in Japan the scientist Kurosawa has discovered this because of the abnormal growth of the rice plants, because of the fungal infection, and the constituents it is secreting is what? Gibberellins. Hundreds of gibberellins were discovered, of which the widely noted is gibberellic acid 3. Okay. What is the precursors of gibberellins? Acetyl coenzyme A and mevalonic acid, and the precursors are terpene precursors. and the derivatives are terpenes. Look at the functions. This will be here only, it will not go anywhere. After class also you can note down, I will give you time. First is seed germination. Okay. Gibberellic acid promotes seed germination. How is it promoting seed germination? Let us take the monocot seed once again. So, we know in a monocot seed, endosperm is present and just a papery cotyledon is present, that is scutellum. This is what ma? Endosperm. You will have starchy endosperm at the center, and all throughout the monocot seed you will be having what? Proteinaceous endosperm. Okay. And here there is an embryonal axis. Towards the tip of the embryonal axis, here you have plumule. Plumule is uncovered by coleoptile, and here it is coleorhiza and radicle. Growth of this embryonal axis only will be after the seed germination as well, right? This will show positive phototrophic movement, this will show positive geotrophic movement. Where is the energy coming from? Not from the cotyledon, it is being extracted from the starchy endosperm. To digest the complex starch, what is the enzyme required? Amylase, yes or no? This amylase synthesis is induced by gibberellic acid. How gibberellic acid is promoting seed germination? Gibberellic acid will induce the synthesis of amylase. Amylase will act on starch, and this will convert it to the simple sugars where embryonal axis can take it. There how the seed germination will happen. Understood everybody? Any doubt anyone? This is the process behind the promotion of seed germination by gibberellic acid. What is the hormone that is inhibiting seed germination? Abscisic acid. Abscisic acid will promote seed dormancy. Okay. Whenever the environmental conditions are not favorable for seed germination, at that point what happens? Abscisic acid will induce seed dormancy. It will inhibit seed germination. So, when seed germination activity is considered, what is that promoting? Gibberellic acid. And what is that inhibiting? Abscisic acid. Both of them are antagonistic to each other. Clear? You have understood about seed germination, ma? Next is bolting. What is bolting? Increase in internodal elongation. When is that happening, and mostly in what type of plants you will see? No, you will mostly observe it in rosetta plants like cauliflower, cabbage. Okay. Yes. In these plants what happens is, just prior to the flowering, just before flowering, gibberellic acids will increase the internodal elongation. Internodal elongation. Just prior to the flowering means just before they are entering to the reproductive phase, the gibberellic acids will increase what? Internodal elongation. When the internodes get elongated, definitely the stems will be elongated. The floral primordia will produce large type of flowers and fruits later. Okay? That is the reason behind increase in internodal elongation prior to flowering. Understood? What is the reason behind, ma? If the internodal elongation happens prior to flowering, after flowering, floral primordia, flowers are formed, thereby vegetables, fruits will be developed. Yes or no? They will be produced in large size. Okay, and they will have enough space to grow. Okay, that is the reason they are sprayed prior to the flowering. Next is stem elongation. Stem elongation, we have seen it in the discovery only, no? Okay, gibberellins will promote the stem elongation by promoting the growth of internodes. And you are clear with seed germination. These three are the major functions of gibberellic acid. Now, how gibberellic acids are applied? How are they used? Okay, they are used in brewing industries. How? Because gibberellic acids will induce the synthesis of amylase, and that is how simple sugars will be formed. That is the reason they are used in what? Brewing industry. They will enhance the sugarcane yield, okay. They will enhance the quicker maturity in conifers, okay, and they delay ripening. So, when ripening is considered, gibberellic acid is inhibiting and ethylene promotes ripening. These both are what ma? Antagonistic in their function, and this will increase the size of fruits as well, okay. This will increase the size of fruit. For example, if the traditional apple size is of this much, let us suppose, but with the action or the spray of gibberellins, they will be increasing in its size to this huge. One small example I have told you, okay. This we will use to increase the grape stocks, so that we will get higher size of grapes. Same the case in mangoes, in bananas, in sugarcane, okay. When stock size increases, it will inhibit, it will be in a position to adapt so many fruits in that, yes or no. So, these are the functions and applications of gibberellins. Clear everybody? And then abscisic acid. Around 1960s, abscisic acid is discovered by 3 different scientists in 3 different ways, like abscissin, dormin and inhibitor. Inhibin 2 or inhibitor, like that 3 different ways, 3 different compositions were discovered. Later they have identified that all 3 are of one phytohormone only, that is abscisic acid, which is activated during stressful conditions. So, this is addressed as stress hormone or anti-transparent hormone. Stress hormone or anti-transparent hormone. Which comes to function during stressful conditions. First activity that it promotes is what ma? Stomatal closure. What is the precursor of abscisic acid? Vialoxanthin. Abscisic acid is composed of what derivatives? Terpenoids. Carotenoids also you can take. Derivatives of carotenoids. So, the first function it initiates is what ma? Stomatal closure. Immediately I already told you, whenever there is stress, it will shut down its doors, that is induced by abscisic acid. Next, it promotes the abscission. It will reduce the burden of the plant, like how I told you. And then seed dormancy, it promotes seed dormancy. When abscission you have to take, this is antagonistic with cytokinin. When seed dormancy you have to take, this is antagonistic to gibberellic or heat stress. Understood? Functions and applications. See, every point I am explaining you clearly. Then and there I am giving you out synergistic and antagonistic function to make it clear. Okay, you will not get beyond this. Every corner I am covering. Okay. Next is ethylene. Auxins, is a scientist who has proved that when when ripened oranges are placed beside the unripe bananas, it also caused the ripening of bananas, which proved that there is some gaseous hormone that is being transmitted from this ripened oranges to unripe bananas, which cause the ripening in them. And later that gaseous hormone is identified as ethylene, which is primarily involved in fruit ripening. Fruit ripening. Okay, and the synthetic form of ethylene that we use is ethaphone. Here we go with the functions of ethylene. What is the precursor of ethylene? Methionine. Okay, and it is derivatives gaseous hydrocarbons. First, it promotes the flowering in pineapple and mangoes as well. Increases fruit ripening. It promotes tripled response in rice seedlings. Promotes senescence, and it promotes the growth of adventitious roots in stem cutting regions. In that case, it is synergistic to auxin. Applications: fruit ripening, abscission and thinning. Thinning of the fruit. See, you see a fresh fruit which is unripe, the wall layers will be thick, but if you see ripened fruits, the walls of the fruit will be thin. Fruit walls gets thin. That thinning is because of what? Ethylene. It increases the fruit set, sprouting of storage organs. That is because of ethylene. These are the applications and functions of ethylene. This is all about phytohormones, ma. Most important synergistic and antagonistic effects you have to remember is first about apical dominance. Two phytohormones are antagonistic. Which promotes apical dominance? Which promotes? Auxins. Which inhibits? Cytokinins. Which promotes seed germination? Seed germination. Gibberellins. Which will inhibit seed germination? Abscisic acid. Which promotes abscission? Abscisic acid. Which inhibits abscission? Cytokinin. Okay. Which promotes senescence? Which phytohormone promotes senescence? Ethylene. Which will inhibit senescence? Cytokinin. So you can note it down later also. After the class I will give you time. Now we will move on solving the objective. Chalo. First question, ma. Team A, first question. Find the correct match. Auxin was isolated by FW Wendt. Miller et al. What is this discovery? Cytokinin. Artificial form of cytokinin, that is kinetin. He has isolated it from autoclaved herring sperm's DNA. Abscisic acid, it is not anti-aging hormone. What is anti-aging hormone? Cytokinin. Ethylene is keratin derivative? No. Methionine. And it is a gaseous hormone. It is not keratin derivative. Clear? Second question, team B. Second question, team B. Phototrophic and geotrophic response towards the source. Phototrophic and geotrophic response towards the source of stimulus. Tell me the answer, ma. Go through the following matches with respect to plant growth regulators and its derivatives. Auxins are indole derivatives, Gibberellins are terpene derivatives, cytokinin adenine derivatives, and the statement given in the NCERT is N6-furyl-aminopurin. Abscisic acid, carotenoid derivatives. So all the above are correct only, right ma? Next question. Next question to team C. Cell division promoting hormone that cannot be found in the plants. Cannot be found in the plants means artificial form. That is how you have to apply. Okay. He is asking cell division promoting hormones means what? Cytokinin, which is not present in plants, cannot be found in plants means artificial form of cytokinin. Next question. First isolated cytokinin of plant origin. Zeatin. How many of the following terms are associated with the stress hormone? Stress hormone, first of all, it is abscisic acid or what derivatives? Terpenoid derivatives or carotenoid, not terpene. Terpene derivatives are gibberellic acid. Yes, dormin. Yes, right, one of the type of discovery, dormin. Not bolting. Bolting is associated with Richmond-Lang effect is associated with cytokinin. Seed dormancy is associated with abscisic acid only. Anti-gibberellic acid is abscisic acid when you consider seed germination. It will not break dormancy, abscisic acid will promote dormancy. So, how many terms are associated with ABA here ma? 3. Got it? This is how you have to choose. I hope you understood everyone. Be little active, louder. Which one is the precursor of ethylene? A. Methionine. 8th question ma? B. These are natural auxins, indole acetic acid and indole butyric acid. What function they do? Parthenocarpy. Their application is parthenocarpy. 9th question. Listen, abscisic acid to acidic in nature. Gibberellin is gibberellic acid, acidic in nature. Auxin, indole derivatives, natural form is indole acetic acid, acidic in nature. So, which is not acid? 10th question beta. Most widely used plant growth regulator in agriculture is characterized by. He is asking plant growth regulator means only 3 hormones, right? What are they? Auxin, cytokinins, gibberellic acid. Now you see. Promotes abscission in flowers and fruits? No. Initiates germination in peanuts? Increases rate of respiration? No. What increases rate of respiration? See, at the type of fruit ripening, okay, respiration rate increases, which is also known as climacteric respiration. Okay. So, what is the right answer? 11th question beta. C, the form, right? Ethylene is not directly applied in the field as a gas due to its high diffusion rate. Which compound is used to overcome this limitation? Ethaform. Next question, 12th question. Find the incorrectly matched. Listen, Auxin promotes flowering in pineapple, Gibberellins foolish seedling disease, Ethylene promotes female flowers in cucumbers, Cytokinin promotes apical dominance. Cytokinin doesn't promote apical dominance. Remember here, I missed out there, Auxin promotes flowering in pineapple, Ethylene also promotes flowering in pineapple and mangoes. Gibberellins, yeah, that is foolish seedling disease. Next, Ethylene promotes female flowers in cucumber, correct only. Incorrect statement is option D. Next, 13th question ma. Richmond-Lang effect. Cytokinin, good. 14th question. Read the following statements. It is used to prepare weed-free lawns. It promotes the abscission of older mature leaves and fruits. So, these functions are with respect to auxin. 15th question. Which of the following does not fall under triple action of ethylene? Triple response we have learned, right? Triple response is horizontal growth and swelling of the stem. Fruit response is not included, ma. See, apical hook formation and swelling of the stem, horizontal growth of the stem. This is promoted. These are the triple responses. So, nowhere fruit response is not included in the triple response. These three are included. Okay. What are they? Horizontal growth, prevents geotropism and swelling of stem. 16th question you look into. Odd one out of the functions. Functions of what? Cytokinin and abscisic acid. It is very clear. Louder? This is incorrect. This promotes seed dormancy and this inhibits seed dormancy. Stomatal opening, stomatal closure. Delays senescence, promotes senescence. Chloroplast formation, destruction of chlorophyll. Next question. Apical bud inhibits the growth of lateral buds. This effect can be counteracted by. 18th question. Most common and more active cytokinin is zeatin. 19th question. Richmond-Lang effect is related with delaying senescence. 20th question ma. You need to identify the correct option with respect to true or false. Okay, fine. This question is about jasminic acid, salicylic acid, not required. Brasinosteroids, not required for you. Till this question, though, it is clear, no ma? Yes? We will solve few more set of questions on cell biology and plant physiology in the coming class. From coming class we are going to start the core botany chapter starting from living world, biological classification, plant kingdom, morphology, anatomy of flowering plants. Will be done around 4 to 5 classes, okay. So, be there. Till here we have completed the major portion of the NCERT level, that is cell biology and plant physiology, wherein from these 6 chapters we can expect 7 to 9 questions. 7 to 9 questions, okay. So, practice more and more objective, do objective oriented preparation, okay. Do not sit with the content all the time, invest more time in the practice, okay. Yeah, continue, and if you are getting any doubts just let me know.